Sun and to the Symposium on Hearing Disorders from Bench to Bedside. This morning we have 3 distinguished speakers, 2 top-notch in the field of hearing research, Paul Fuchs and Jeff Holt, and then a rising talent from here, from Argentina, Maria Eugenia Gomez Casati. And we organized this meeting together with Juan Goutman, and it's called, it's sponsored by the Association for Research in Otolaryngology, ARO, which is the main hearing association in the US and I guess worldwide. And they funded us to bring both Paul and Jeff here. So it's called the ARO Mini Symposium. And it is a pleasure to have 2 distinguished speakers, foreign speakers back in Argentina, and 2 friends. And I'm only going to introduce Paul today, and Juan is going to introduce Maru and Jeff. And I'm not going to give a spoiler for Maru nor Jeff. Paul Fuchs is a Rubinstein professor of otolaryngology, head and neck surgery at Hopkins University School of Medicine, and also has appointment at the bioengineering and neuroscience departments at Hopkins. And he's also co-director of the Center for Sensory Biology. But above all, Paul is a friend of the house. I was thinking last night, I think this is the 5th time Paul has come to Argentina, and probably the 3rd time to San and Taller. Those who have my age might remember Paul dancing the entire night. And there's Lydia, the entire night. Well, yeah, but that was before when he started working with Leach, yeah. So Paul used to dance a lot during our Taller meetings. Then he came to Falan. Then he came invited by Osvaldo Chitel for a neuroscience course. And I think this is the 5th time he is here. He's a great friend and collaborator of our lab. I think I met Paul in '95 when immediately after finishing my postdoc. We met at an ARO meeting in Tampa, and Paul was working in a synapse in the ear where I had cloned a receptor. And since then, we've been collaborating first with an ARO grant, and for the last 20 years, we've had an NIH grant together. And people from my lab has gone to his lab to get trained, especially Eleonora, who has been instrumental in establishing electrophysiology in the inner ear in Argentina. So I think that our lab has a important space within the hearing field worldwide. And Paul has been, Paul's help and collaboration has been instrumental for the development of hearing research here in Argentina. And for that, we are very thankful, Paul. Paul has made seminal contributions to synaptic physiology in the ear. All synapses that are present within the cochlea, both afferent and efferent synapses, he has studied, and his work together with Elizabeth Glowatsky in his lab have made seminal contributions. And Paul is going to talk today about, give us some insight into the efferent system within the inner ear. So Paul, the stage is yours.
Okay, muchas gracias, buenos dias. That's all my Spanglish. So from now on, we're done. And so my purpose this morning is twofold. First, to start my timer, to make sure I don't abuse the system here. Okay. My purpose this morning is twofold. I want to give you a sort of background into how the periphery of the auditory system is regulated by cholinergic feedback from the central nervous system. So this is among the many fascinating aspects of how the ear works. And I'm going to be setting the stage for a subsequent talk by Maru with that introduction as well. And perhaps a little bit of setting the stage for Jeff Holt's talk. I'll very briefly say something about how mechanotransduction fits into the cochlea. But my other purpose is then to go on to some recent work we've done where we've tried to solve the problem of visualizing nicotinic acetylcholine receptors in the cochlea. And these have been very resistant to the usual strategies of antibody labeling. And so I'll show you what our alternatives have been. So yes, moving. Oh, so this picture that you're looking at now sort of makes the point that the cochlea receives a very potent efferent innervation. We're looking down on a hole mount where you see the hair cells are kind of red and the efferent synapses are labeled by an antibody to choline acetyltransferase. So you see these big green blobs on the bottom of what are called the outer hair cells. That's the most potent form of feedback inhibition. There's also a nexus of cholinergic synapses underneath the inner hair cells. That's the single row on the bottom that you see down there. And so to go on then, I'm going to tell you about how the cholinergic receptors fit into this picture. So first, the big picture about how the cochlea works. This is a kind of schematic that points out that as sound comes into the ear through the external and middle ears, it sets up a frequency-dependent pattern of vibration in the cochlea called the traveling wave, first described by von Bekeschi back in the 1930s. Now, this traveling wave is a pure kind of physical phenomenon, but it's exaggerated, amplified, and improved by a positive feedback mechanism involving mechanical amplification provided by the outer hair cells, and I'll explain that in detail. That process results in a larger and more frequency selective vibration pattern, which then stimulates inner hair cells, which are the principal source of afferent innervation to the brain. So 95% of all the afferents contact inner hair cells, send information to the brain, where it does a whole lot of stuff like giving us the percept of sound, but it also activates efferent neurons located in the brainstem near the superior olivary complex. And those feed back. There's inhibitory synaptic contacts onto outer hair cells from the medial olivocochlear efferents, and I'll talk exclusively about those today. There are also lateral olivocochlear efferents, which form synaptic contacts on the afferents below the inner hair cells. We know less about their functional role. They're more difficult to study, but for today, we're going to focus just on these guys here that make they inhibit the outer hair cells. Okay. All right. So how do we know about this system? Well, historically, what happened first was that a guy named Rasmussen did degeneration studies where he cut the 8th nerve. Then he looked for degenerating cell bodies, and he found them in the region of the superior olivary complex. So they must have axons then that were cut by this operation. Subsequently, Paul Guth and others then showed that those same cell bodies were cholinergic using choline histochemistry. And the great experiment was done by Bob Galambos in the 1950s, who put an electrode into the 8th nerve of an animal under anesthesia. He presented a sound, recorded the compound action potential elicited by that sound, and then combined that with electrical stimulation of the efferent axons in the floor of the 4th ventricle. And what he found was that when he combined his acoustic stimulus with this electrical shock, the compound action potential was made smaller. So definite proof that this is a negative feedback process. That was elaborated on by many people after that time of Galambos, but mostly using his technique of electrical stimulation of the efferent axons in the floor of the 4th ventricle. So Bob Wiederhold and Nelson Kiang show the same effect here at the level of a single afferent neuron being driven by an acoustic stimulus. Its firing rate is almost 100 spikes per second. During efferent stimulation, it almost entirely shuts down. Now, a much more intriguing observation was made by John Guinan and colleagues, who then asked, well, how does this efferent feedback vary as a function of the frequency content of the sound? So the classical way to describe the tuning of a cochlear afferent nerve is to ask, how sensitive is it to sounds of different frequencies and intensities? And you thereby establish this threshold tuning curve that you see here, a very sharp V-shaped tuning curve. This says that this particular individual afferent is most sensitive at this frequency, and then it takes louder and louder sound to produce a response at other frequencies. Now, when you do the same kind of tuning curve description, in the presence of electrical stimulation, the efferents, you see a very interesting thing happening. Yes, the neuron is inhibited at its characteristic or best frequency, but off that frequency, it's not changed. So what inhibition does is not only desensitize the cochlea, but it detunes the cochlea. It disrupts or degrades the cochlear frequency selectivity mechanism. Well, how does that happen? Particularly because I've already told you that this afferent neuron contacts an inner hair cell on this side of the cochlear epithelium, but the inhibition that we're talking about is delivered to these outer hair cells, which themselves have very, very sparse and really not very significant afferent contact. So here, inhibition to outer hair cells is changing the response of the inner hair cell. So the answer lies in the fact that I've already talked about in the big picture slide. That is that there's a tuned mechanical vibration called the traveling wave, which propagates along the length of the cochlea, varying at its point of maximal vibration according to the mechanical properties of the basilar membrane. Near the base of the cochlea, at the bottom of the snail shell, the membrane is very stiff, so it responds best to high frequencies. As you propagate and travel out to the apex of the cochlea, the membrane becomes less and less stiff, more floppy, and so it responds better to lower frequency vibration. And so the traveling wave for a lower frequency tone propagates to near the apex of the cochlea, reaching its maximum there. But a higher frequency wave propagates less far and rises to a peak at a point where the mechanical properties of the basilar membrane are appropriate for that frequency of sound. Now, if you draw the envelope of that traveling wave, what you see is that it sort of has two components. It has this lower, broader shoulder down here, and then this very sharp peak at the characteristic frequency for that position. This so-called passive mechanism is the response of a dead cochlea. So even if the animal is no longer alive, you still get this pattern of tuning. But when the animal is alive, there's some biologically sensitive energy-requiring mechanism, which I'm going to tell you is an active mechanical amplification provided by the outer hair cells. This is a mechanism called electromotility, and it results from the following. The hair cell has mechanotransduction that Jeff Holt's going to tell you about mechanotransduction in much more detail. This sets up a change in the membrane potential of the hair cell, and that consequently causes changes in the shape of voltage-dependent proteins called prestin. So they undergo a conformational change, which is voltage-driven, and there are hundreds and hundreds of copies of prestin in the basolateral membrane of the hair cell. The combined force of all those conformational changes leads to a macroscopic change in the shape of the outer hair cell. So when the outer hair cell is depolarized during the acoustic stimulus, it gets shorter, and when it's hyperpolarized, it gets longer. This then gives rise to an enhanced vibration pattern across all of the membranes. So the traveling wave is a longitudinal sort of wave, but it also has a kind of a lateral rocking motion, such that there's a lateral shear developed between the underlying sensory epithelium and the overlying tectorial membrane, an acellular gelatinous sheet, which contacts the stereocilia of the hair cells and pushes them from side to side. That's the adequate stimulus to cause a change in membrane potential. Okay, we're frozen. Thank you. All right, look at that, it's really moving. All right, so then if we look in more detail, the point being now that as the sound wave comes in and exerts an excitatory stimulus, that's pushing the hair bundles to the taller hairs, that causes the outer hair cells to depolarize, and so they then shorten a bit and increase the overall deflection of these membranes, thereby increasing the stimulus being delivered to the inner hair cell. Now, this is a somewhat exaggerated drawing, except actually at threshold, most of the movement is provided by the outer hair cells, not the incoming sound wave. So if you get rid of the outer hair cells, you become 100 to almost 1,000-fold less sensitive to sound. So this is really an important mechanism for setting up the fundamental sensitivity and frequency selectivity of the cochlea. So now you can imagine, okay, this is a voltage-dependent process. If you synaptically inhibit the outer hair cells, then maybe you're going to interrupt this electromotility, and that indeed is the case. So here's what the traveling wave, oops, sorry. Come back, little Sheba. There you go. All right. So this is what the original traveling wave looks like. This is what the traveling wave looks like in the presence of efferent inhibition. So a blunting of the peak here, but it doesn't change this part, the low passive part. And so if you just turn this upside down in your mind's eye, you can see that that really looks like the tuning of the afferent fiber in control and during inhibition. So we think this is really the explanation for how the efferents do regulate the sensitivity and tuning of the cochlea by inhibiting the outer hair cells, damping their mechanical amplification. One other thing about these efferent neurons is that they themselves respond to sound. So Don Robertson and colleagues did a really nice experiment where they recorded from individual efferent neurons in the brainstem and presented sound. And in the same animal, they also recorded from afferent neurons. So here are the afferent tuning curves. We've already seen some of these nice V-shaped afferent tuning curves. But here's the efferents. And you can see that their tuning is almost as good as that of the afferents. In other words, they are very sensitive to a narrow band of frequencies. So the efferents are frequency tuned, and they are very sensitive. They have a high sensitivity. In fact, their firing rate varies almost linearly as a function of the intensity of the sound. So here's the action potential frequency as a function of sound intensity. And this is from work from the Liberman laboratory. So this means that these efferents are frequency and intensity tuned. So their activity, their feedback to suppress the sensitivity of the cochlea is going to vary as a function of what the acoustic environment has in it. So particular frequencies that are the loudest are going to be most inhibited, et cetera, so that this feedback mechanism is a way to enhance your sensitivity to particular signals in a complex acoustic environment. All right, so how does this inhibition happen at a cellular level? Well, now I'm going to turn away from the mammalian cochlea and tell you that the original discoveries here were all done on non-mammalian vertebrates. And that's okay, because so far, every place that a hair cell has been examined, whether it's in a fish or a fowl or a frog or a turtle, you name it, or a mammal, the mechanism seems to be universal. So this is a highly conserved mechanism of inhibition of these cells. So the first really telling experiment was done by Robert Fettyplace and Andrew Crawford and their colleagues, where they made intracellular recordings from auditory hair cells in the turtle ear. And yes, turtles can hear, and they have feedback inhibition. And so the average change in membrane potential to a single shock to the efferent axons produces this biphasic effect. You see there's a small depolarization and this much larger hyperpolarization that lasts for almost 100 milliseconds. Now, this is membrane potential relative to the resting membrane potential of about minus 50 millivolts. So those of you that are electrophysiologically minded will recognize that this is starting to get close to the potassium equilibrium potential, and is probably unlikely to be due to a chloride conductance change. And indeed, we now know from a whole host of different kinds of experiments, including molecular experiments from Belén's lab, that this mechanism is a two-channel synaptic inhibition where there's an acetylcholine receptor, which is itself a ligand-gated cation channel with high calcium permeability. So calcium enters through the acetylcholine receptor. It then leads to the activation of nearby calcium-dependent potassium channels. So that's what this big hyperpolarization is due to. In most cases, these are the small conductance class of calcium-activated potassium channels. So this two-channel mechanism was first observed in turtle, but understanding how it came about depended on some other work. And so here's some experiments from chicken hair cells, which were isolated from the auditory epithelium and then recorded from with gigome seal whole cell recording pipettes. So in an isolated chicken hair cell like this, with this kind of arrangement, you can record either membrane potential or membrane current, and you can stimulate the efferent effect by applying acetylcholine from a nearby puffer pipette. So there's no efferent synapse here, it's just acetylcholine. And what you get in terms of the membrane potential is this biphasic change that looks very similar in overall shape to the native efferent synaptic potentials in the turtle. This effect now we know is due to acetylcholine alone. It's not like these efferents have two neurotransmitters, one of which is excitatory and one's inhibitory. That's ruled out. It's all acetylcholine. Furthermore, there's probably not an electrical synapse and a chemical synapse. All of those other alternative hypotheses are excluded. And if you take that same cell and put it into voltage clamp, what you can show is that there's a biphasic change in membrane current, inward and outward current, as you might expect from the hypothesis. And furthermore, now this also says that there's not some complex voltage-dependent conductance which is activated subsequent to a ligand-gated channel. Furthermore, if you load such a cell with a strong calcium buffer like BAPTA, which you can do through your patch pipette, then you eliminate this outward current, showing that it is indeed a calcium-dependent current, and the remaining current is a small inward current, as we see here. So these kinds of experiments and many others in which different components are knocked out, et cetera, have all been done to confirm that this is really true for, as I said, all hair cells that have been examined to date. If you record from a hair cell and it has an efferent synapse, it works like that. And so Belen and her colleagues were really responsible then for nailing down the molecular biology of the acetylcholine receptor expressed by hair cells. They identified these two genes, which I'm sure you've heard about from them, alpha 9 and alpha 10, members of the bigger family of ligand-binding alpha subunits of the nicotinic acetylcholine receptor family. They're somewhat related to the muscle receptor in being sensitive to alpha-bunga toxin, but they have rather bizarre pharmacology overall. For example, they're not activated by nicotine, which is a bit of an odd thing, but nonetheless the case that these are really ligand-gated cation channels. They probably have a pentameric structure like this, where 2 alpha subunits are the ligand-binding elements, and 3 alpha 9 subunits, and 3 alpha 10 subunits probably act more like beta subunits. So this is the receptor. And now that we have this molecular biology, we can do all kinds of cool experiments, some of which Maru is going to tell you about in the subsequent talk. But what I want to do now is turn to a remaining problem which has bedeviled people in this field for a long time. And that is that how can we visualize these receptors? I mean, we can study them physiologically, we can study them with molecular biology. It's kind of tough to show where they are expressed. As I said at the very outset, we and many others have really failed to generate reliable antibodies to label these receptors in situ. So how can we visualize them by other means? And we've explored 2 alternatives. One is to use a peptide toxin, which is conjugated with a fluorophore. I'll tell you more about that in a moment. And then the other is to do genetically conjugated biomarkers. So use genetics to attach something that you can visualize to the receptor. So what we did was to collaborate with this wonderful guy, Michael McIntosh at the University of Utah, who studies the protein components of cone snail venom. Now, you may know about cone snails. These are carnivorous snails that capture fish and eat them. And they do that by shooting a poison-laden harpoon into the fish. And that harpoon contains a wide variety of bioactive peptides, including among them one which binds very tightly and very selectively to alpha 9 containing receptors. Interesting. So what they have been able to do then in Michael McIntosh's lab is to chemically alter that peptide a bit. It's called RGIA. They made a chemical modification that makes it RGIA 5727. And then with that chemical modification, they could attach a Cy3 fluorophore. Now, all of Michael McIntosh's work is based on heterologous expression of recombinant receptors in xenophus oocytes. So first off, we wanted to know, okay, does the same thing apply to a native receptor in a cell that we know expresses these receptors, that is the hair cell? And so Yanyan Zhang in my laboratory did recordings from hair cells and then applied a 10 nanomolar concentration of Cy3 RGIA. So this is the fluorophore conjugated form. And it gradually reduces the synaptic current recorded in an inner hair cell being stimulated by electrical shocks to the efferent axons. So this is work now in hair cells of the mouse. So we're back to mammals now. These are all inward going currents because we're negative to the potassium equilibrium potential. So potassium current is inward going. So the block comes on. It takes 20 minutes or so to produce almost complete block. And then it just doesn't wash off. So this is an intracellular recording. You know, they're not going to last forever, but within 45 minutes, we could get no reversal. The rate of onset of the block is shown over here, and it varies somewhat according to the conditions of the tissue. What we did find, which was quite helpful, is that this peptide by itself is already rather sticky, But once it's conjugated with a fluorophore, it becomes even stickier to tissue. So it tends to congregate in different spaces in the tissue, to percolate through the tissue very slowly. But if you pre-expose the tissue to bovine serum albumin, what you do is load up all those extracellular charged sites. And so then the toxin gets in a little more expeditiously, as shown here by this green line. So within a matter of a few minutes. And you can get almost complete block under those conditions, whereas it's somewhat more difficult to get total block. And this takes a while to do, but we know that you can still get good synaptic recordings in control cells that last for an equally long period of time. Okay, so it works. This conotoxin still acts like a blocker of alpha-9 containing receptors. And the important thing is, does it visualize the receptors? And the answer to that is yes, very nicely. Again, this is work from Yan-Yang Zhang, and now with the help of Philippe Vincent in the laboratory. And what they found is that if you look at the cochlea, almost invisible up here. Okay, if you look at the cochlea, I won't use that one. This is a whole mount. So this is where you see the hair cells labeled green by incorporation of a fluorescent dye that enters the hair cell through the mechanotransduction channel. So it turns them green. And then the Cy3 conjugated peptide toxin is shown in red. And down here, the combined image shows the 3 rows of outer hair cells and a single row of inner hair cells with these little red blobs at the bottom, which are where they should be if you're labeling acetylcholine receptors in these cells. Now, those of you with a good memory may see that I previously didn't show any synapses on inner hair cells in my schematic, but in early postnatal development, there's this strange thing that happens. So for about the first 2 weeks after birth in a rodent, those efferent synapses are also on the inner hair cells, and they work the same way as the efferent synapses on the outer hair cells, but then after about 2 weeks, they disappear. So this is some interesting kind of developmental process that we still are exploring the meaning of. But what it means is that if you look at a young hair cell, a young cochlea like this one, which is a P10 mouse, you are going to see efferent synapses on both outer and inner hair cells. And if you do the control experiment, so this is an alpha-9 knockout mouse, it doesn't have any alpha-9 receptors, there's no labeling, that's great. If you pre-expose the tissue to unlabeled conotoxin and fill up all the binding sites, then you also don't get any specific labeling. Now, this allows me then to point out a couple of weaknesses of this technique. Although it's pretty good, one of the problems is that, as I mentioned, this stuff is rather sticky. So it tends to congregate and get stuck in places where you don't expect there to be synapses, like in this region medial to the inner hair cells. There's just diffusional trapping of the protein, of the peptide. The other more serious problem, serious limitation, is that it only works on live tissue. So if you fix the tissue first, no binding. If you apply the toxin and then fix the tissue, it kills the fluorescence. So we haven't solved that problem yet. But if you have an application where you want to look at the expression of alpha-9 containing receptors in live tissue, this is a good way to go. But it is limiting. So we wanted to do another thing. And what we did was, I'm just going to jump over that for now. We went to a genetic tool. So Hakim Hayal learned how to do CRISPR-Cas9 modification. And using that, he was able to attach a hemagglutinin tag to the cytoplasmic tail, the C-terminus tail of the alpha-9 or the alpha-10 proteins. So he made both alpha-9 HA tag mice and alpha-10 HA tag mice. And the PCR proof of the binding is down here for the homozygous, heterozygous, and wild type condition. It works. And the hearing is normal. That's good. We didn't want to mess up their ears. Too bad. So they have thresholds which are as good as that of wild type. So down here are shown acoustic brainstem response thresholds. You present a sound, you measure the evoked response in the brain, and what you get are, you can measure the threshold to sound. Wild type, heterozygous, and homozygous animals for both alpha-9 tagged and alpha-10 tagged are identical to the wild type. So that's good. Their hearing's normal. Furthermore, the waveform of the evoked response, this auditory brainstem response, looks identical in both wild type, heterozygous, and homozygous. So that's a good thing too. And the labeling works. So we can visualize these receptors in the tissue. So here what is shown are whole mount views of the cochleas where the efferent terminals now are labeled with a protein that binds to a presynaptic, with an antibody that binds to a presynaptic protein. That's SV2 in red. The HA tag is shown in green, and the nuclei of the hair cells are made blue by DAPI. So here's the 3 rows of outer hair cells. There's a single row of inner hair cells. This is in I think this is a 3-week-old animal. It may say it on the slide somewhere, but it's about 3 weeks old. So you don't expect any labeling on the inner hair cells. And you can see that the heterozygous condition, the homozygous condition are both equally good. So that's nice. We can see, we can use the heterozygotes, for example, then to label and maybe do other manipulations as well. And there's no labeling in the wild type animal as shown over here. So that's for the alpha 9 tagged mouse, and this is for the alpha 10 tagged mouse, and the same story applies. We get really nice labeling that corresponds to the efferent innervation, again, with in this case, with choline acetyltransferase antibodies for the efferents. And what Hakim did in this case was to look along the length of the cochlea from the basalmost turn, the highest frequency region, to the middle turn, to the apical turn, and you can see that there's pretty decent labeling throughout the length of the cochlea. So that all works pretty well. So we have a good way to label, and we can use the heterozygotes as well as the homozygotes. Now, what about the synapse? Obviously, it's going to be important to know whether or not you still get activation of the acetylcholine receptor when it's got this HA tag stuck out there on the C terminus. And the answer is, for the most part, everything's okay. So these are recordings that Yuanyuan Zhang did from outer hair cells, where she stimulated the efferents here with a shock, and then under voltage clamp can produce outward currents at minus 44 millivolts, that is positive to the potassium equilibrium potential, or inward currents negative to the potassium equilibrium potential. So we think it's still the same biphasic response. And this is for the heterozygous condition for alpha 9. This is for the homozygous condition for alpha 9. Down here is shown the data then for the alpha 10 animals, where again, in the heterozygous, they're really not different from wild type. And here the homozygotes are giving us a hint that there's something odd maybe going on for the homozygous condition. And so what I'll tell you is that averaging across all the recordings that we've done, the postsynaptic response, if you just measure the amplitude of the postsynaptic response, it looks pretty good in wild type, heterozygous, and homozygous, really not very different. But weirdly, the probability of release seems to be down in the homozygous condition compared to the heterozygous condition. So something is kind of providing a retrograde effect on the efferent terminals such that the probability of transmitter release is lower in the homozygous HA tagged animals. Now, we don't really know why, but Megan Wood did some nice quantitative immunochemistry to look at the synapses on the outer hair cells. And what she found was that the number of synaptic contacts was unchanged. So there's usually 2 to 3 efferent synapses on each outer hair cell. That was not altered. But when she looked at the volume of those efferent synapses, what she saw was that there was a significant difference in the volume. So for the homozygous condition in both the alpha 9 homozygote and the alpha 10 homozygote, the total volume of the synaptic contact was down. Now, we don't really know if that explains anything, but at least it says there's something going on in which the structure and function of the efferent terminals is being affected by this acetylcholine receptor with an HA tag in the homozygous condition. But that's okay, because we know the labeling is great in the heterozygote. In fact, it's just as good. So we would say that if you want to use this animal model to study alpha 9 or alpha 10 containing receptors, you want to use the heterozygotes, because we think the synapses are working normally under that condition. And so that's that. I want to now thank the people that did all the hard work: Hakim Hayel, Fatima Shakir, Yuanyuan Zhang, Philippe Vincent, and Megan Wood. We've had wonderful collaborators, most recently Michael McIntosh, whom I mentioned. But then, of course, I think it's 20 plus years of collaboration with Belen Elgoyhen and her lab. She helped us to design the viral vector to make the HA tags. And thank you very much for your attention. Happy to take questions if there are any. Lydia looks like she has one. I'll repeat the question. Go ahead.
To hear cells frequencies sensitive. So I don't understand if this is because of there is a gradient of electrophysiological characteristics along the cochlea, or because the relationship with the basilar membrane changes along the cochlea?
Thank you. That's a really great question. So what Lydia is asking is, the outer hair cells have a sort of a characteristic frequency. Is that just because of where they sit in the cochlea, or do their electrophysiological properties also do something to give them tuning? And the answer is both are important. So if you look at the size of the outer hair cells along the length of the cochlea, in the low frequency end, the outer hair cells are long. And as you go toward the high frequency end, they get shorter and shorter and shorter. So the whole thing looks like an organ pipe array. It's extraordinary. So that means that the membrane time constant goes shorter and shorter and shorter as you go to higher and higher frequencies. The conductance, the number of channels per unit area, goes up, so that this little tiny high frequency outer hair cell has a larger macroscopic conductance than this great big one down in the apex. So very short membrane time constant. That's a good thing. And there's also something really cool about the way the adaptation of the mechanotransduction process occurs as well, which also goes toward giving them a sort of electrical tuning. It's not as wonderful as it happens in non-mammalian vertebrates, but there's something there. Thank you.
Hi, Paul. Great talk. I'm wondering if you see the expression, the levels of expression of alpha 9, alpha 10 with the level in the outer hair cells.
I didn't quite get that.
If you study the level of the expression in alpha 9, alpha 10 when you when you
Oh.
put the tag,
Yeah, no.
because you
We
you
didn't try to
say-
quantify anything yet. No, we just looked to see if it's in the right place.
Okay,
So we didn't measure amounts at all.
okay.
Yeah, that would be cool though, because we have a good tag and we can, if you want to get enough tissue, you could get it out.
Yes, I wonder if the level affect your volume of the synapse or no.
Ah, okay. Interesting idea, but we don't know yet. And I don't want to isolate 10,000 cochleas to get enough tissue.
Yeah, it's very difficult.
Hi, thank you for your nice talk. I was wondering, so I heard a lot about the Synapse from Belen's group, and I'm always curious to think to know what you think about why you need acetylcholine inhibition instead of having GABA, for example. Is it a kinetics difference? So why to bother to have a calcium-dependent potassium?
Yeah. Okay, I'll give you my half-baked, terrible evolutionary story if you have a minute. Okay, so if you look at the vestibular epithelia, there's efferent feedback, which is predominantly excitatory. So that's mediated by sort of more normal kind of neuronal nicotinic receptors that cause excitation of the afferent neurons. So efferent feedback to the vestibular epithelia has an inhibitory component, but it's mostly excitatory. So my idea about this is that during evolution, neurons split off from the facial motor nucleus, and you can see that embryologically, that's where they come from, these efferents. They split off from the facial motor nucleus and projected out to the cochlea. Now, they were motor neurons, so they were cholinergic. And so initially, they were used to cause excitation of the vestibular epithelium, which is important because otherwise, the vestibular epithelium can saturate in the negative context. So as you rotate your head from side to side, you have negative cutoff on one side as you rotate. So if you can boost it up out of that, you have a preservation of dynamic range by excitation. So that's a good evolutionary argument for why you might want efferent excitatory feedback to the vestibule. But then somewhere along the line, those efferents started contacting the hair cells, and that elicited a different form of response. And the outer hair cell synapse, the hair cell synapse is really unusual. It's got a near membrane cistern, looks like a little calcium store sitting right close to the membrane where the efferent synapse is formed. So that somehow there was a mistake made during evolution that added those potassium channels. So it was an excitatory synapse. And now you have this kind of calcium-dependent inhibition that acts there. So why they didn't use GABA? Because they didn't have GABA. They had acetylcholine, so they did what they could with what they had. That's my potted evolutionary explanation.
Okay. So thank you, Paul.
Thank you very much for your attention.
Now we move to the second part.
Now we move to the second talk, which is Maru Gomez. Okay, second talk is going to be Maru Gomez Casati. It's my honor, pleasure to present her. She's not only a great scientist, she's also my friend, I would say. So I would only say a few words about Maru. Maru studied actually in Rosario and moved to Buenos Aires to do her PhD together with Belén and Eleonora Katz. And she did great work here in Buenos Aires, then moved to Boston, to Harvard Medical School, to the lab of Gabriel Corfas for a postdoc. She stayed there for a few years. She got a Pew Fellowship for that. So she returned with that help to set up her own lab at the School of Medicine, University of Buenos Aires, Institute of Pharmacology, where she's now working on the things that she will talk about today. So about how noise produces damage to the cochlea, and also how aging produces changes to our hearing capacities. And thanks, Maru, for talking to us.
Well, thank you a lot for your nice introduction. I would like to thank all the organizers. It's a pleasure for me to be part of this symposium with these two great researchers. So, and thank you, Paul, for your nice introduction about the Efferent system. So I can go directly to results. So our lab is trying to understand the mechanism behind noise-induced and age-related hearing loss, and to study the role of the medial olivocochlear efferent system in protecting the cochlea. And also, we are trying to understand the role of this system in the development of the auditory pathway, with the final goal to translate this knowledge into therapeutic approaches to prevent or treat hearing loss. So in the mature organ of Corti, inner hair cells are mainly innervated by around 10 to 20 auditory nerve fibers or afferent fibers. These synapses are glutamatergic and mediated by AMPA receptors. Outer hair cells receive efferent contact from the medial olivocochlear system, and that make direct synaptic contact with outer hair cells. As already Paul says, activation of this MOC pathway reduces the cochlear sensitivity to the action of alpha-9, alpha-10 receptors in outer hair cells coupled with calcium-dependent potassium channels. So activation of this MOC pathway reduces the cochlear sensitivity. So one important question in auditory neuroscience is what role this MOC feedback plays in our ability to hear. It has been proposed that it controls the dynamic range of hearing, improves signal detection in background noise, mediates selective attention, protect the inner ear from acoustic injury, prevent age-related hearing loss, and shape the normal cochlear development. Today, I will be talking about these three last topics in the role of the MOC system during noise-induced age-related hearing loss, and some preliminary data about the role of this system during the development of the auditory system. So every day we experience noise in our environment. Noise exposure of above 85 decibels can be dangerous. It can damage sensitive structure in the inner ear and cause noise-induced hearing loss. Depending on the intensity and the duration of the sounds, the changes can be permanent or temporary. Permanent damage is associated with outer hair cell death, with afferent terminal degeneration, leading to auditory thresholds elevation. While temporary hearing loss is associated with dislocalization of the stereocilia of the cells and reversible damage in the synaptic terminals. It has recently been shown by Charlie Lieberman's lab that noise exposure that didn't cause permanent auditory thresholds elevation can still cause a permanent damage. As you see here, there's the auditory brainstem responses thresholds at different frequency tests, and they exposed mice, adult mice, to noise of 100 decibels for 1 hour, as shown here in black. Here is the auditory thresholds at different frequencies, and 1 day after exposure, they observed that the thresholds were elevated. However, 7 days after, they returned to normal. That means that the threshold shift was transient. However, when they look at the first peak of the ABR, they observe a permanent reduction in this peak. This peak represents the sum activity induced by sound at the first synapse between inner hair cells and auditory nerve fibers. As I already told you, inner hair cells receive between 10 to 20 synaptic contacts from afferent neurons. Each synapse has a special pre- and post-synaptic specialization. So if we label the pre-synapse with one antibody CTBP2 revive, we can label the presynaptic machinery in these cells. And also we can use one antibody for AMPA-type glutamate receptor, the GLUAR2 subunit antibody that recognizes the AMPA receptor, and we can label the postsynaptic element as you see in green. In the normal ear, there is a perfect colocalization between pre- and postsynaptic elements. That means that the amount of inner hair cell synapses can be counted at the confocal microscope. And as you can see here, there's the labeling of the inner hair cell with myosin, and you can see that you have between the 10 to 20 synaptic contacts. However, after exposing mice to this moderate noise that didn't cause permanent threshold elevation, they observed a 40% reduction in the number of synaptic contacts in the inner hair cells. And this reduction in the synaptic contacts correlates with the synapse, with the reduction in the peak 1 amplitude. So they discover for the first time that exposure to noise, to transient noise, is more dangerous than we thought. And because of the normal audiogram, it has been called hidden hearing loss, because it cannot be tested with the normal audiometry. So the results I'm going to show you today have been done by Luis Boero and Valeria Castaña. And we have been working with the different mouse model of MOC inhibition. So we use to test the role of the MOC system in this hidden hearing loss. So we use an alpha-9 subunit knockout mice that lacks cholinergic transmission between MOC fibers and outer hair cells. And we have an alpha-9 knocking gain of function mice that has a point mutation in the alpha-9 subunit. This mice was developed in Belén Elgoyhen's lab, and it has a point mutation that enhances the activity of this receptor. So as is shown in the diagram, we have wild-type mice with normal MOC activity, alpha-9 knockout with no MOC activity, and the knock-ins with one enhanced MOC activity. So we first tested the auditory function in the different genotypes at P21. So we first measured the ABR thresholds at different frequencies, and we observed no difference between the wild types and in black and the alpha-9 knockouts in gray, except at the highest frequency in the ABR thresholds. However, we observed a 5 to 10 dB elevation in the knock-ins. We then measured the ABR peak 1 amplitude, and we didn't observe any difference in this peak 1 amplitude that, remember, represent the sum activity at the first synapse between inner hair cells and auditory nerve fibers. We then measured distortion product of acoustic emission that this type of recording, we can have one idea of the outer hair cell function, so the electromotility of the cell. If we apply 2 tones of very close frequency to the cochlea because of the electromotility of these outer hair cells, we can record sound in the ear, and we can perform like in ABR threshold and a curve at different test frequencies. And this type of recording reflects if we have, if the outer hair cells are functioning properly. So we did the DP recordings, and we observed no difference between wild types and the knockouts, and we observed a 5 to 10 dB elevations in the thresholds in the knock-ins that has an enhanced MOC function. This cochlear elevation, whether measured by ABR or by DP, were the same. It was from 5 to 10 dB and reflect an strength MOC activity due to the enhancement of alpha-9 and alpha-10 receptor activity. Because it has been shown by Belen's lab that if we block alpha-9, alpha-10 receptor in the knock-ins with strychnine, that is an antagonist of this receptor, auditory thresholds return to normal. So we then tested what happened if we expose these mice to moderate noise levels that cause only transient thresholds elevation. So to do that, we exposed the 3 genotypes to a noise level of 100 decibels for 1 hour. And we observed that in wild-type mice, we have the next day after exposure, we have a threshold elevation that returns to normal after 7 days. That is a clear sign of mice that has a phenotype of hidden loss. In contrast, in the knockout with no MOC activity, we observe after 7 days of exposure a permanent elevation. So that means that the damage is worse comparing to wild types. And interestingly, mice with enhanced MOC function were completely resistant to the same noise protocol. We then measured the peak 1 amplitude of the ABR as a sign of cochlear synaptopathy, and we observed after 7 days of exposure a permanent reduction in the peak 1 amplitude, both in wild types and in alpha-9 knockouts, but in the knock-ins, there was no modification of the ABR peak 1 amplitude at all the frequency tested, suggesting that the enhancement of the MOC system can prevent the loss of ribbon synapse. So to test that, we did the whole mount immune staining organ of corti at different cochlear regions, and we used the antibodies that I told you before, CTBP2 in red that labels the presynaptic ribbon and GluO2 to label the postsynaptic PANTA. And we count those PANTA that co-localize as a synapse. So we did that in control animals and at 7 days after acoustic exposure. As you can see here in the quantification, we observed that after acoustic trauma, 7 days after acoustic trauma, there was a reduction in both the pre, post, and co-localized synaptic PANTA, both in wild types and in alpha-9 knockouts. However, in mice with enhanced MOC function, we found that after 7 days of acoustic exposure, there was bigger counts of both pre, post, and co-localized synaptic PANTA. That result was very surprising, and it suggests that activation of the MOC system can not only prevent the loss of ribbon synapses, but also it may be involved in synapse formation. We then test the function of the outer hair cell by recording distortion product of the acoustic emission, and we observe the same trend as ABR recording. So we observe that depending of the MOC level of activity, the damage was worse. So we observe an inverse correlation. So in wild-type mice, the DP thresholds were transient. In the knockouts, after 7 days of acoustic exposure, the threshold levels remain elevated. And interestingly, in the knock-ins, they were completely resistant to the same noise protocol. We then tested, as already Paul mentioned, we did a whole mountain immunostaining to observe and quantify the MOC terminals under the outer hair cells, and to study if exposure to this level of noise caused any change in these MOC terminals. As you can see in wild types, we did the immunostaining with synaptophysin, and as you can see in wild types, in the base of the outer hair cells, there's these efferent contacts, and that typically occur as clusters under the outer hair cells. As you can see in black, typically outer hair cells are innervated by around 1 to 5 MOC terminals. And 7 days after acoustic exposure, we observed in wild types that there was a reduction in the number of MOC terminals after noise trauma. And we observed that most of the outer hair cells most of the innervation occurs as singlets rather than in cluster. And most of the outer hair cells were innervated by 1 to 2 terminals. We did the same experiment in the alpha-9 knockouts, and we observed that in the knockout, without being in control conditions, they have lower MOC terminals in the outer hair cell. Most of the outer hair cells were innervated by 1 to 2 terminal, and there was no modification after acoustic trauma. In the knock-ins, it happened the contrary, that in control situation, outer hair cells were already innervated by more MOC terminal, reaching like some of them were innervating with 6 to 7 efferent terminals. So as a summary of this first part, we can say that enhancement of the MOC system prevent the loss of cochlear synaptopathy or loss of ribbon synapses, and that impairment of this system can lead to more severe consequences after acoustic overexposure. And we propose that both cochlear synaptopathy, so the loss of ribbon synapses, and the loss of MOC terminals contribute to the hidden hearing loss phenotypes, compromising the performance of complex to hear, for example, speech in noisy environment. So our next question was, can potentiation of the MOC system prevent or delay age-related hearing loss? To do that, we use wild types and alpha-9 knock-ins at 6 months and 1 year of age. And we have these mice in a sound control room at the animal facility, so they were never exposed to high level of sound. We measure the ABRs, as you can see here in wild types in black. We measure the ABRs at different frequencies, and we observe that at 1 year of age, there was an elevation in the ABR thresholds. In contrast, in the knock-ins, there was no modification at 1 year of age. We then measured the distortion product of the acoustic emission, and we observed the same trend. In aged mice, so at 1 year of age, there was an elevation in the DP thresholds, and there was no modification in the threshold in mice with enhanced MOC function. We then measured the ABR peak 1 amplitude as a sign of cochlear synaptopathy, and we observed that wild type is in black and alpha-9 knock-ins in red. We observed that at 6 months of age, there was no difference in the ABR peak 1 amplitude at all the frequencies tested. However, at 1 year of age, there was a reduction in the peak 1 amplitude in the wild types that were more evident in the high frequency regions. So we did the same whole mount immune staining using CTBP2 and GluO2, and already, as I already told you, we count those panta that colocalize as a synapse. We did the experiments in wild type and in the knock-ins at 6 months of age, and we didn't observe any difference in the amount, in the density of synapse in inner hair cells. However, when we did the same cochlear immunostaining at 1 year of age, we observed a significant reduction only in the wild types. As you can see here, the reduction was at all the region in the cochleas of the cochlea, and it was more pronounced at higher frequencies, indicating that enhancement of the MOC system in this mice can prevent the loss of synapses. So we then did the whole mount immunostaining using NFH and DRAG5. NFH labels all afferent and efferent fibers in the cochlea, and DRAG5 is to label the nucleus of the cells. And we didn't observe any difference between wild type and the knock-ins in the global efferent and afferent innervation. However, when we count the outer hair cells, we observe a small but significant reduction in the number of outer hair cells in wild types compared to the knock-ins. That means that the enhancement of the MOC system, so activation of the alpha-9, alpha-10 receptor, can protect from the outer hair cell loss due to aging. So as a summary of this part, we can say that enhancement of the MOC system prevent the age-related cochlear synaptopathy, have a protective effect against outer hair cell damage, and that potentiation of this reflex delays presbyacusis. These results are very promising in order to find drugs that potentiate alpha-9, alpha-10 receptor to treat or prevent hearing loss. And the last part, I will talk about preliminary data that was done by Valeria Castaña, working on the role of the MOC system during development of the auditory pathway. As Paul already have told you, in inner hair cells of artificial mammals, before the onset of hearing, display spontaneous activity, and it has been shown that during this critical period of development, before the onset of hearing, there's descending MOC fiber making direct contact to inner hair cells, and this synapse is mediated, as I already told you, is the same synapse as in the outer hair cells, but in the immature epithelium. And this synapse is mediated by alpha-9, alpha-10 receptor coupled to SK2 channels. So it has been proposed that this spontaneous activity is very critical and important for the development of the auditory pathway. So, and that this synaptic inhibition modulates this activity and is also critical for this development. So what we did is we tracked the auditory onset in alpha-9 and in the alpha-9 and alpha-10 knockout, and we observed that the onset of hearing is around P12, P13 in mice, and we find that in mice with enhanced MOC function, as you can see here in red, they start to hear between 1 to 2 days before their wild types. So we start having ABR recordings as soon as at P13, and they reach the mature thresholds levels 1 to 2 days before the wild type. And in contrast, the onset of hearing in mice with in the alpha-9 knockout mice was delayed comparing to the wild types. And the same happened with the distortion product of acoustic emission, suggesting that the transient efferent inhibition to inner hair cells is very important to establish the maturation of the immature auditory system. We then measure the ABR peak 1 amplitude, and also we measure the amplitude of peak 2 that represents the relay at the cochlear nucleus level, and we also measure the ABR peak 3 amplitude that represents the superior olivary complex. And we tracked the maximal response amplitude for these first 3 peaks, ABR peaks, and at different postnatal ages. And we normalize this maximal response amplitude to the peak, the corresponding peak amplitude at P21, that is a mature state. And we observed that at P16, when auditory thresholds reach the same levels in the 3 genotypes, there was no difference in the ABR peak 1 amplitude. However, both peak 2 and peak 3, they didn't reach the mature levels in the 3 genotypes. That means that cochlear maturation begins in the periphery and goes beyond the onset of hearing in the different relays at the central nervous system. So we really wanted to know what happened if we expose mice to the same noise level that we did at P21 as soon as they start to hear at P14. And so what we did the same noise protocol as I described before of 100 decibels for 1 hour, and we exposed wild types, alpha-9 knockouts, and the knock-ins. And we were surprised to see that in the wild types, 1 day after acoustic trauma, there was threshold elevation, But 7 days after, this threshold didn't return to normal like we observed at P21. And that means that noise exposure during this critical period of development had permanent, at least at 7 days. We are testing to longer ages, but at least at 7 days, it caused more permanent damage to the cochlea. We observed the same with the ABR peak 1 amplitude at 7 days after acoustic coma. They didn't recover, and it happened the same with the alpha 9 knockout. However, the alpha 9 knocking, we didn't observe any difference neither in the ABR threshold or the peak 1 amplitude at different frequencies. So that means that this result shows that the auditory sensitivity in mice with enhanced or new MOC activity are altered at hearing onset, that maturation of the central connections begin in the periphery and continues after the onset of hearing in central relays. And we observe that mice exposed at this critical period of development have enhanced sensitivity to permanent cochlear damage. So I would like to thank the organizers, the collaborators, and also Luis and Valeria Castaña that did all the job. Thanks a lot.
Thank you very much, Mario. We have time for a couple of questions. Jeff.
Thank you. Very nice talk, Meru. So I think it might not be practical to make alpha 9 knock in humans. And I'm wondering if there's other ways you're thinking about boosting cholinergic signaling in the MOCs that could be therapeutically practical.
I think, well, the next step is trying to find a positive allosteric modulator that we can potentiate alpha 9 and alpha 10 receptor as a possible therapy for hearing loss. The only problem is that alpha 9, alpha 10 receptor, as already Paul says, has a very peculiar pharmacology, and most of the nicotinic agonists are antagonists on alpha 9, alpha 10 receptor. In fact, nicotine is an antagonist. So until now, Belén's lab only find 3 drugs that potentiate alpha 9, alpha 10 receptor, that is ascorbic acid, ryanodine, and one chloroviguadine is one agonist of serotonin receptor, chlorophenylbiguanidine, I don't know how to pronounce. Only these 3 receptors. So the ideal thing will be to find some specific site of interaction, and if we can find some positive modulator without any side effect, that will be perfect.
Yeah, thank you. Thank you. Very beautiful work. So quick question. Are alpha 9, probably I should know this by hearing all your talks, but I forgot. Are alpha 9 and alpha 10 somewhere else in the brain, not only in the cochlea? And if it is in the pathway below, is it involved in some sort of plasticity during development?
Well, alpha 9 and alpha 10 receptors are in the vestibular system. It has been found them in lymphocytes, right? Lymphocytes and the dorsal root ganglion neurons. But there's one piece of work that says that they are in the brain, but Belén, during her postdoc, never found alpha 9, alpha 10 in the brain. And in the alpha 9, alpha 10 tag mouse, there's no expression in the brain. So there's only, I think that work from Barbara Morley, she did antibody work, but they are not expressed, at least in our hands, Belén and Paul's. there's no expression at the central level. Yeah.
Okay, we need to move. Thank Maru again for your talk. Great talk. We will move to our third speaker, Jeff Holt, who is professor of otolaryngology in Harvard Medical School. Jeff did his PhD in the University of Rochester, if I'm not wrong, already working in inner ear in potassium channels, and then moved to study the mechanotransduction mechanism in the cochlea, in the lab of David Corey, already again in Harvard Medical School. And so I just wanted to say that this is a very difficult topic for the auditory science, because it's difficult to, only a handful of labs around the world can actually, have the technique to evaluate in a very, biophysically sophisticated method the mechanotransduction. So it's the process by which the ear can actually transform, convert, transduce actually mechanical stimuli, sounds into electrical potentials. And that takes a lot of skills. So helped made a lot of discoveries in that field. He then established his own lab in 2001, I think, in the University of Virginia first, not only working on that topic, but also developing top tools, viral tools to express, to re-establish auditory function in animal models that have mutations in this in the mechanotransduction apparatus. And in the last few years, he's been primarily involved in identifying the actual molecule that is responsible for sound detection in this mechanotransduction system. And I should also mention that molecule is it's a mechanosensitive channel as expected. And similar molecules have been identified for other senses throughout the last decades, but only in the last few years, Jeff and his colleagues have been able to identify which is the molecule that is responsible for this sound detection in the hearing system, obviously. So it's a great pleasure to have you here, and the stage is yours.
Wonderful. Thank you, Juan, and thank you, Belen, for the invitations. I'm really happy to be here. I've been talking to Juan and Belen for a number of years, wanting to come visit Buenos Aires, and this is my first time here. So I'm thrilled to be here. And I also want to say that I'm a big soccer fan. I wish Argentina good luck in the World Cup coming up. I'll be cheering for you. Okay, so yes, my lab studies the sensory hair cells of the inner ear. We've already heard a couple of beautiful talks about these hair cells, but we are interested in understanding this mechanotransduction process, how these hair bundles wiggle back and forth and convert sound into electrical signals. And in particular, we wanted to identify the molecules involved. So I'm going to tell you about a family of proteins that we discovered their function. There's 8 of these. They're known as TMCs. That stands for Transmembrane Channel. They're numbered TMCs 1 through 8, and 2 of them are expressed in the ear, TMCs 1 and 2. Interestingly, during development, there's a switch in expression in the cochlea. So TMC2 is expressed first and transiently in the cochlea. Its expression declines, and then TMC1 expression rises and lasts into adulthood. So these sensory hair cells, of course, reside within the inner ear. In the human ear, there's about 16,000 sensory hair cells in the cochlea and about 30,000 hair cells within the vestibular organs. And so in the human, these are not easily accessible. We can't really see them. They're embedded in the densest bone of the body, the temporal bone. So instead, we ended up using the mouse as a model system. And this is an image I borrowed from Doris Wu. It's a quite beautiful paint fill, but it shows these inner ear structures right here. So we can dissect out the cochlea from the mouse and then look at this image in a scanning electron micrograph. And here you can see, again, the hair cells that we've already heard about from Paul and Meru. one row of inner hair cells and 3 rows of outer hair cells. And you can see their cell bodies here. I've just colored in blue. And if we zoom in on a single hair bundle right here, this is the typical sort of staircase array, and they're organized in this V-shape arrangement. So it's movement of this hair bundle that then gets converted into electrical signals. And so electrophysiologically, we can measure this. Here we've got a single hair bundle. Most of the cell body is out of the image. And if we place a recording pipette on the cell body and at the same time wiggle this hair bundle back and forth, we can plot the position of the hair bundle, that's shown here above, and measure the current that flows into the cell. So when the hair bundle swings to the right towards its tallest stereocilium, that's a positive deflection, and that evokes a large inward current of several hundred picoamps. When the bundle swings to the left, we consider that an inhibitory stimulus, a negative stimulus, and the current level drops back to 0. So we've been interested to really understand this at the molecular level. How is it that motion of this hair bundle structure is converted into these electrical signals? And the idea was originally proposed by David Corey and Jim Hudspeth about 40 years ago. They suggested that there may be a series of mechanically sensitive ion channels here at the tips that could be modulated. Their open probability could be modulated with this motion. And so if I blow up a single pair here on the right, some of the basic elements of this mechanosensory transduction apparatus are shown right here. And so the thinking is when this bundle is deflected, there's an increased tension in this structure right here known as a tip link, and that will physically pull open this mechanosensitive ion channel, allowing cations to flow into the cell, primarily calcium and potassium. And so work over the past couple of decades has identified some of these molecules. The tip link itself is composed of cadherin 23 and protocadherin 15. And those are also Usher syndrome proteins. So we know that mutation in those lead to hearing loss and vision loss. The upper and lower ends of this are thought to be myosin molecules, and they perform an adaptation function. I'm not going to talk about that too much, but these myosins can anchor to this actin core. At the upper end, we think that's myosin 7A, also an Usher syndrome protein causing deafness and blindness. But this mechanosensitive ion channel has really been a major question mark in the field for at least the past 40 years. And so we've been interested to identify that. It's been of great interest, and there's been a number of publications over the years suggesting ion channel candidates. I just list up a partial list right here, the original citation, but it seems like one after another, the profile of these was raised, but then subsequent publications showed that they were not involved, either not expressed in the ear or not expressed in humans, not expressed in mammals, or knockout of those genes did not lead to any auditory phenotype. But coming back to the one we were interested in, we looked at this list and decided that this one, TMC1, was worth a closer look. And I don't have time to really go into all of the evidence over the past 10 years, but we now have a pretty significant body of evidence suggesting that indeed TMC1 is forming the hair cell transduction channel. And I think one of the key publications was this. It was a collaboration, and 3 postdocs who contributed to this work, 2 from my lab and 1 from David Corey's lab. And let me just spend a few minutes talking about some of the highlights from this publication from a couple years ago. So one of the first things we did, because we did not have a structure for this protein at the time, was to see if we could generate a model. And so we used a couple of computer programs to predict a structure based on homology with another known structure from TMEM16A. And this is what the computer, this was actually work done by Marco Sotomayor at Ohio State using the supercomputer. And so the computer spit out this as a prediction. It suggested that there were 2 proteins coming together as a dimer. Each monomer contained 10 transmembrane domains, and there was a putative pore-forming region of this. And so we weren't sure if this was just some computer fantasy that had been dreamed up or if this had any basis in reality. And so we wanted to test this structure, and so we designed a set of experiments to look into this in more detail. we looked at 17 different sites within the TMC1 protein, really trying to find the pore region itself. And we introduced mutations one at a time. We used a cysteine substitution, so removing the native amino acid, replacing it with a cysteine. And we chose sites here in 4 different transmembrane domains. If I zoom in, you can see those here. Transmembrane domain 4, 5, 6, and 7. And these were based on known deafness sites in humans. These sites right here cause deafness. Some of the adjacent residues we thought were worth looking at. There were some negatively charged residues in this region that we thought might help stabilize cations if they're flowing through an ion channel pore. Some were based on the homology with this other TMEM16A protein that I mentioned, and then one negative control out here in transmembrane domain 8. And so then the idea was to introduce these different mutant forms of the TMC1 using viral vectors, AAVs, and we put them into mice that lacked native TMC1. So these were knockouts for TMC1 and 2. We were able to, oh, that doesn't show up too well, but we were able to record these mechanosensory transduction currents from over 500 hair cells. And that one shows up, yes, 500 hair cells. Uh-oh, we're getting a pause. You guys see that back there? Yeah. Okay, maybe we're back. So we then used brief application of what's known as a cysteine modification reagent. And so the experiment was this: we recorded from the individual hair cell, wiggled the hair bundle, and then puffed on a reagent known as MTSCT. And the chemistry is this. So if you have a cysteine amino acid, this has a sulfur side chain. And if that sulfur side chain is projecting into an aqueous solution, the drug here, MTSCT, can be washed on. It also contains a sulfur side group, and these can form a disulfide bond. And if that occurs within the permeation pathway of an ion channel, you can see how this bulky side group might inhibit the flow of ions. So here's what the data actually looked like experimentally. We gave a repeating series of hair bundle deflections and then washed on our drug right here and recorded these responses. So these are individual hair cell transduction currents. And then as soon as the drug hits the hair bundle, you can see the amplitudes are reduced significantly. When the drug washes out, we didn't actually see any recovery of the currents because we think this was a covalent bond and you couldn't recover from that. This was very robust and repeatable. Here's just a different cell showing the same kind of effect. And then a couple of control traces. So this is with one of the cysteine modifications, but in the absence of the drug, just a control puff, and we see no real change here. And then one more control where this was a wild type TMC1 with the drug, and we saw no real change in the amplitude. So it really depended on having the two together, the cysteine modification as well as the drug being washed on to see these changes in real time. And so we did this for all 17 of the different mutations that were introduced, and here's just a sampling of the data. So the gray shaded region is where the drug was washed on, and so this one, G411, mutated to a cysteine, showed a decline, as you can see here. M412 showed a decline. Some showed very large, rapid declines. Some didn't really change at all. And so we can see several examples, and we analyzed these data, just taking the initial peaks versus the end, and took the current ratio, and we found 5 different sites that showed a reduction in the current amplitude. So because current flowing is a key signature of an ion channel, we really felt like these data were pointing to the idea that these particular sites were likely lining the ion channel pore. Another thing we looked at was calcium selectivity. So I told you earlier that calcium is permeable to these mechanosensory transduction channels. And so in this case, we would wash on a high calcium solution and then measure the reversal potentials both before and after application of the drug. And so this is a lot of data summarized here in this bar graph, and I'm not going to go through all of it, but let me just tell you that there were a number of sites, 7 different sites where the mutation itself caused a reduction in calcium selectivity, and another 11 sites where there was application of the drug caused a reduction of calcium sensitivity. So together, we had mutations that were affecting the currents flowing through the transduction channels, and some sites where there was a reduction in calcium selectivity, key properties of an ion channel protein. So this really led us to the conclusion that these domains we looked at, the transmembrane domains S4 through 7, were lining the ion channel pore. And so again, these are the sites that were reactive. I've just highlighted here. And if we come back to the computer model, we can look at that and map these different sites onto the structure shown right here. And so this would be the outside of the cell. This is the inside of the cell. And we think S4, 5, 6, and 7 are the transmembrane domains that line the ion channel pore. And if I put this in motion, let's see if that's going to work. Is that going to work? No, not going to work. Well, you would have seen the structure rotating, and it just helps you illuminate where the ion channel pathway is. Okay, so we conclude from these data that TMC1 is indeed a pore-forming subunit of the hair cell transduction channel. And another line of evidence also coming from my colleague Marco Sotomayor was a molecular dynamic simulation. So this is also the TMC1 protein embedded in a membrane. And here he's got ions on the inside and outside of the cell, outside and inside. And this movie is, this one's sort of plain, a little glitchy, but what you can see is the potassium ions in red can move down through the ion channel pathway. So our data were strongly supportive of this. And I just want to highlight another publication that came out a couple years ago. Another group identified a method for purifying TMCs 1 and 2 and expressing them in a very reduced system. So this is just protein and lipid. And they were able to reconstitute this and then record from these liposomes. And they were able to evoke single channel currents with a little bit of membrane stretch. They could get a mechanically activated current. So also very, very lovely data. And then most recently, this is a publication that has come out in BioRx as a preprint from Eric Guo's lab. So they have identified a structure for the C. elegans TMC1, and that's shown just here. So the structure based on cryo-EM analysis was very similar to that of ours, shown right here, and another group, Angela Ballesteros. And so I think we can erase this question mark once and for all, and we are now confident that TMC1 is forming the hair cell transduction channel. And so we've been interested in this, not just from a basic science perspective. That's been hugely exciting and satisfying to be part of this work. But we're also interested in it because there are a number of mutations, genetic mutations, that cause hearing loss in humans. And so on the gene structure for TMC1, I've mapped those out here, and all of those shown in black are mutated sites that cause recessive hearing loss in humans. There's 3 or 4 sites shown here in red that cause a dominant form of hearing loss. And so we've been thinking about how we might be able to address this and perhaps attempt to restore function for patients who have genetic hearing loss. And so the strategy is a gene therapy approach where we can use gene replacement. And in general, the goal of gene replacement is if there's a mutated gene that leads to a non-functional protein and a disease phenotype, if we can introduce the healthy DNA sequence using a viral vector, we can perhaps generate the healthy functional protein and lead to correction of that disease phenotype. And so we've been working on this for a number of years. And we have a series of publications where we have sort of iteratively improved the outcomes. And I'd just like to walk you through some of the highlights from these publications. One of the key innovations, I think, was identifying a viral vector that was particularly efficient at targeting the sensory hair cells of the inner ear. And so our current favorite viral capsid is known as AAV9 PHPB, and we used it carrying just green fluorescent protein to illuminate hair cells. We inject this into the mouse cochlea, and you can see a number of hair cells transduced within the spiral of the mouse cochlea. If we zoom in right here, you can see that in higher magnification. So 100% of the inner hair cells are targeted by this vector, and 90 to 95% of the outer hair cells are also targeted. So we think this may be a very efficient vector for carrying DNA sequences into the inner ear. This has also been used by colleagues of ours in Boston in a non-human primate cochlea. So injecting the same vector, they see very robust viral transduction throughout the cochlea in the apex, mid, and basal regions. And we've recently done this in human tissue, not injected into human ears, but in tissue that has been removed from human patients during biopsies. And so applying this in vitro, we can keep the human tissue alive in addition, apply the viral vectors, and we see that the phalloidin here is labeling hair cells, and so we see a lot of hair cells transduced with the same capsid. So we think this may actually be translational and could be useful in the clinic. But more than just carrying green fluorescent protein, we wanted to carry our therapeutic gene. So using the wild type form of TMC1 in this case, and this is in TMC knockout mice, so they lack mechanotransduction completely. This is with bundle stimulation. We see no responses in the knockout mice. But if we inject our AAV vectors with TMC1, we can see robust mechanotransduction currents recovered at the single cell level. And we find that the sensitivity of these cells is similar to those of wild type. So that's reassuring that we can see this recovery of mechanosensory responses. If we look at the basal end of the cell, where the synapses occur, we actually find that in mice that carry TMC1 mutations, there's a loss of synapses in these cells. And so when we inject our vectors, we wanted to see if there was any recovery. And so in black, we see the normal numbers of synapses. And in blue, this is when TMCs are knocked out, there's a significant loss of synapses. But with our viral vectors, we can begin to recover the number of synapses that these hair cells are exhibiting. Furthermore, if we look at their auditory sensitivity of these animals, we see there's a very strong correlation between the number of synapses and the auditory threshold. So softer and softer sounds, well, with more synapses, we're able to detect softer sounds. So we're recovering hair bundle function, synaptic function. What about at the next level of the whole cochlea? So you've already heard about auditory brainstem responses. And so using scalp electrodes, recording the activity of the 8th cranial nerve and the brainstem nuclei to louder and louder sounds on this axis. So for the TMC mutant animals, these flat lines indicate these animals are profoundly deaf. On the right, these are wild type traces. And so this animal, the minimum threshold is about 25, 30 decibels. You can see larger responses for louder sounds. And if we take the knockout mice, introduce our vectors, we see that we can recover these thresholds, in some cases equivalent to those of wild type. So we've got hair cell recovery, synaptic recovery, and whole cochlea recovery. Now, auditory information will rise through the ascending auditory pathways all the way up to cortex. And so we wanted to see if we could get recovery of function in the auditory cortex as well. So collaborating with Dan Pauly at Mass Eye and Ear, we used mice that were injected with our TMC1 vectors. First, this is a control data set. Now here we're looking at each spot is an individual spike from a single auditory neuron. And with louder sounds being played right here, you begin to see spikes that are coincident in time with the sound stimulus. In a TMC animal that's deaf, there's a lot of random spiking, but nothing that's correlated with the sound stimulus. But after introduction of our viral vectors, we see that we can begin to get some of these cortical neurons to respond to sound again. And so over 400 neurons were recorded for this particular study, and we see the tuning curves shown over here. So in gray are individual neurons from normal hearing mice, and in green are cortical neurons from our TMC mutant mice that have been injected with our gene therapies. And so in general, you see that the best responders tended to be clustered around 8 kilohertz or so. And some of them had thresholds around 20 to 30 decibels. Most of them were clustered up here around 60 decibels. But one thing important to notice is the shape of these tuning curves was quite consistent and very similar in both cases between the wild type and recovered mice. So we've been able to look at recovery of function across the ascending auditory system, but is this actually functionally or behaviorally relevant? And to do this in the mouse, I can't really ask the mouse if they can hear. Instead, well, I can ask, but they don't respond usually. Instead, we use an auditory startle response. And so like you, if you play a sudden loud sound for a mouse, the mouse will jump and be startled. And so we can measure the amplitude of their startle response. And so shown here on the right is the responses of wild type mice in black. The TMC mutant animals are completely deaf and they do not respond to a startle stimulus. But after injection of these animals with our gene therapy, we see that their behavioral startle responses is recovered. So the gene therapy approaches over the years, I said we've published several papers on this. This was our auditory thresholds in the very first case back in 2015. Again, the deaf animals shown here in black lines. Here, these had elevated thresholds, but it was taking a mouse that was completely deaf and giving it some auditory function. And it was interesting because my basic science colleagues were very excited about this, but my clinical colleagues said, this is useless. This isn't going to do anybody any good. And so we went back to the drawing board and tried to improve this further. And so we've used several different viral vectors over the years where we've been able to get better responses. I hope my data are going to show up. Well, part of my data show up. But here's the most recent data set. So coming just this year, we've improved our viral vectors to the extent that we now have wild type level responses. And coming back to my basic science colleagues, I've talked to them and they said, Jeff, you already did this a bunch of years ago. Why are you doing this now? But my clinical colleagues see this now and they're very excited. So I think we're headed in the right direction, trying to get this into the clinic. And I'll just summarize to say that we think that TMC1 gene replacement can restore sensory transduction, can restore synapses, can also restore auditory brainstem responses. I didn't show you this, but we do get restoration of DPOAE responses as well. We can recover responses in cortical neurons, as well as auditory startle responses. And so what are the next steps? Well, we think we have what is our clinical candidate, the viral vector with a human TMC1 that we can introduce into human ears. But before we do that, we need to do some pharmacology and toxicology study just to confirm that this is going to be safe. Of course, we're also interested in taking it to the next step, filing an IND application with the US FDA, and then we can perhaps after that, if it's approved, begin clinical trials. I would hope within the next couple of years. So stay tuned. I'm optimistic about this, but I hope this strategy may one day be able to help patients with genetic hearing loss. So I will stop there and just offer some acknowledgements to the folks on my team who have contributed to this work. In particular, I'd like to point out Irina Markovic, who trained here in Buenos Aires with Belén Elgoyhen. She's now a postdoc in my lab, and she did some of the most recent gene therapy work that I presented there at the end. And great. Thank you for your attention. Happy to take any questions.
For questions, Jeff, you said that you would support Argentina in the World Cup?
I'm sorry?
You said that you would support Argentina in the World Cup, the soccer World Cup?
Yes, I do.
I can get you in trouble at home, right?
Yeah.
Because his wife is French.
Beautiful work, Jeff. I have a couple of questions first. The gene therapy was done in young mice or adult mice? I mean, that's the first. The second, is there any gene therapy already in clinical trials for restoration of hearing loss? And the third question is, I mean, there's several mutations in TMC that lead to deafness in humans. The idea is to do knocking or to restore their mutations, or for all mutations just replace the wild type TMC.
Okay, three good questions. The first one about the young mice. Yes, these injections were done during the first postnatal week. We would like to be able to do it in mature mice, but we just find that technically quite difficult. The bone becomes ossified and it's hard to access that inner ear space without doing damage. We've been trying to perfect that technique, but so far haven't had success. What might limit that is we do know that TMC mutations will eventually lead to hair cell death, and you would need to have surviving hair cells for this therapy to work. So there may be some developmental limit on when it would be effective. What I can say is some of the mutations are milder in humans and some are more severe, and maybe this partly addresses your third question. I think all of the recessive mutations, and there's about 65 of those identified in TMC1, could potentially be targeted with this strategy. We tried it for dominant mutations and it does not work there. Overexpressing the wild type does not overcome the dominant mutations, but for the rest of the recessive mutations, I think it could work. Some are severe, profound loss of function mutations, and those may be the hardest to overcome. Some are milder mutations, and I think that may allow for an expanded capacity for recovery. Let's see, your second question was FDA, or are there any
clinical trials?
Are there any approved clinical trials for hearing loss? Not
Not
yet.
approved, but in clinical trials. Yeah.
Yeah, so there's a couple companies in the US that are interested in this, and one of them just 2 weeks ago got the okay from the FDA to begin a clinical trial for otoferlin mutations.
Okay.
Otoferlin is one of the synaptic proteins, and it's expressed in inner hair cells.
But concerning my third question, the gene therapy would be with the wild type TMC, not to restore each mutation. I mean, because there are several mutations in TMC that lead to hearing loss.
Right.
So the therapy would be just wild type TMC.
Yes, the therapy would be to use the wild type sequence, and I think that could
Yeah.
be a benefit for perhaps all 65 of the recessive
Okay.
mutations.
Okay, thank you.
Yes.
I have two questions. The first one is a more evolutionary oriented question regarding TMC. So it's known that potassium in the conditions of the cochlea produces a depolarization, which is clearly, when you teach this to the students, creates problems. But so you said that the TMC channel leads potassium and calcium. And so the question is, does it lead sodium too, or there is no sodium conductivity? And the second question, is there any known role for calcium in the TMC, in the cochlea?
Yes, so
that
good
pathway.
questions. This is a non-selective cation channel. So experimentally, when we do this work, our extracellular solution includes sodium, potassium, and calcium. And we see evidence that all three can permeate the channel. In the native environment, the endolymphatic solutions that bathe the hair bundle contain primarily potassium and low concentrations of calcium. And so to answer your question about calcium, calcium does permeate the channel, and it seems to have a few different roles in cells, in hair cells. As a second messenger, one of the prominent things is it contributes to adaptation. So if you deflect a hair bundle, there is a decline in the response. You may have seen some of that in my data, but that decline in the response, we think, is due to a calcium-dependent adaptation process.
So it's a fast adapting response.
Yeah.
And the second question is more regarding your own work, is the TMC, the deaf mice through TMC knockout, do they suffer from any other problem rather in
Yeah.
addition to being deaf?
Right, good question. So for TMC1 mutations, that only seems to cause deafness. TMC2, I said, is expressed transiently in the cochlea. Turns out in the vestibular organs, it's expressed into mature stages. And so if you have mutations in TMC2, that seems to cause vestibular dysfunction. So they are both involved in the inner ear, but to our knowledge, nowhere else in the body. We haven't seen them anywhere else, and we haven't seen any other phenotypes associated with mutations in TMCs 1 or 2. Yes.
Beautiful work. Just one question. How long does it last, the injection, or does it last forever, the gene therapy?
Yeah, great question. And that's something we would like to have a long-term answer to. So far, I can tell you we've gone out to 1 year of age and the effect seems to be stable. In other gene therapy applications, in the retina, for example, there's been work done in dogs where injection of gene therapy into the retina has persisted for up to 10 years. So it may be a long-term effect in the ear as well. We don't have the data yet. With one injection, yes.
Any other question? If not, we thank Jeff
Okay,
again.
thanks very much.
Thanks to all the speakers again. Thank you all for listening. And yeah, we'll just move on, maybe.
Well, we have a coffee break now at the Ifebine, and then we wait you back here at in half an hour, sorry.